I'm delighted to be here today to talk about MetaZooGene, a working group of the Scientific Committee on Oceanic Research, or SCORE, formed in 2019. Goals are reflected in our title, a new global view of marine zooplankton biodiversity based on DNA metabarcoding and reference DNA sequence databases. MetaZooGene Working Group 157 now includes 23 members from 19 countries. The SCORE mission of international cooperation is actually fundamental for MetaZooGene, and it is consistent with our goal of characterizing patterns of pelagic biodiversity throughout the ocean. Our primary goal, primary function is integrative molecular morphological taxonomic analysis, and I'll talk more about those methods in a moment. Our term of reference is to create an open access web portal for DNA barcodes, and also to design the best practices, they're called, usually optimal DNA barcoding pipeline, best practices for DNA metabarcoding. But in fact, we're currently discussing approaches for intercomparison or intercollaboration. There's just too much variation for us to ever hope for a single best practice. The scientific mission of MetaZooGene is founded in DNA barcoding of marine zooplankton. Barcodes are DNA sequences determined from morphologically identified specimens. The usual barcode gene is mitochondrial CO1, showing patterns of variation that can discriminate species across the zooplankton assemblage, shown here on the left. Another gene used to trace the evolution of life on Earth is the tree of life gene, or 18S ribosomal RNA. A short hypervariable region is frequently used as a barcode to classify major groups of taxonomic zooplankton, but it's not sufficiently variable to identify species reliably. The molecular world was transformed with the advent of high throughput DNA sequencing in the early 2000s. Whole genome sequencing got cheaper and faster, and a subsequent application involved analysis of environmental samples for short sequences, barcodes, which is called metabarcoding. Now, this technology relies upon high-throughput DNA sequencing, yields millions of DNA sequences, allows large-scale analysis of complex environmental samples, but the data analysis, bioinformatics, is extremely challenging and complicated, and that's hence one of the mission areas for MetaZooGene. Now, as I mentioned, a primary goal for MetaZooGene is to a new reference database. Now, the MetaZooGene Barcode Atlas and Database is a product of Todd O'Brien. He's a MetaZooGene member from NOAA Fisheries in the US, and he's basically the creator of this new reference database, which is meant to provide much better capacity for identification of species and taxonomic groups of marine zooplankton. Based upon CO1, we now have more than 200,000 CO1 sequences for more than 11,000 species of marine zooplankton. And notice that number is bigger than 8,000 holozooplankton I mentioned. We also do include merozooplankton, benthic adults. We also include the tree of life gene, 18S RNA, and Todd has recently added fish and protists. The database is searchable by taxonomic group and ocean region, and Todd has also created a special form of the MetaZooGene, which is specific to the ICES North Atlantic and inland seas, in partnership with the ICES Working Group for Integrative Morphological Molecular Taxonomy, or WIGIMT. I'll say more about that WIGIMT partnership in a moment. MetaZooGene also released the announcement of our new database in a review paper in Marine Biology. This is open access and provides a current summary of CO1 barcoding for marine zooplankton. Another product from MetaZooGene recently was a 2021 themed set of the IC's Journal of Marine Sciences, Patterns of Biodiversity of Marine Zooplankton based on molecular analysis, which was Editor-in-Chief Howard Brauman assisted with. The papers were by MetaZooGene authors and others on a topic near and dear to our heart. Then in this year, MetaZooGene has been named as a partner, a UN Ocean Decade partner in association with Marine Life 2030. It's a wonderful opportunity for us to expand our impact and also our outreach. I'm here partly today because we will host a symposium on Friday here, New Insights into Biodiversity, Biogeography, Ecology, Evolution of Marine Zooplankton based on molecular approaches, co-sponsored between SCORE, MetaZooGene, and ICES. So it's another partnership activity. Pre-registration was required. I'd like to acknowledge all of the MetaZooGene working group members, as well as our partners with ICES Working Group for Integrative Morphological Molecular Taxonomy, WIGIMT, and our foundations in the Census of Marine Life program, Census of Marine Zooplankton from 2005 to 2010. Thank you for your attention.